Violeta's name because I thought Violeta will present.
Okay. We are from the same team, so I'm happy to represent Impact Hub Athens and Remedies for this presentation. How many minutes do I have for now, Risa?
You have 10 to 15 minutes.
Okay. I'll try to keep it tight. Welcome everybody. Thank you for the invitation of the cluster. I would like to say a few words first about the Impact Hub Athens and then our participation in Remedies. So Impact Hub Athens is building communities for impact. So we are part of a bigger network of 100 impact hubs around the world, where we see that we need to prototype and to find solutions in emerging challenges around the environment and society. So we believe that the pathway is to work with communities, to engage communities and to have different approaches and mobilize resources in order to be able to tackle collectively those challenges. In our journey, we have been participating to Remedies. Is a project that started already since 4 years. So we are now in the last year of operating, and we decided to tackle plastic pollution. That's why we call it Remedies. We work around detection and monitoring, collection and valorization, but also prevention and zero waste solutions. So it's a lighthouse project of the Mission Ocean. We are a group of different partners. I will present you in a while. And today we will present you like the solutions, like the technologies and the solutions that we develop within the consortium in the different demo sites that we operate. So from monitoring to remediation. The Remedies, as I said, is co-founded by the European Commission. We co-create solutions for the future of our oceans. We deploy micro and plastic litter valorization and prevention pathways. We are part of a bigger mission. We want to say that we create a movement, or we contribute to a movement, one act at a time. We are acting from the individual to the organization and to the collective, and we want to bring on the board all the different stakeholders who have experience or they have the wish to contribute for a plastic waste free ecosystem. We come together to change our habits and co-create a plastic conscious society. As I said, we come from different areas across the Mediterranean, but also from the north. As you can see, we are 23 partners from 12 countries, and we collectively join the forces to find solutions for the Mediterranean. Here is like our distributed demo sites. We have a variety of locations that we can find similar patterns, but also common challenges and common solutions that they can probably be replicated to the different demo sites across the Med. Again, being connected with our pillars, this is like our focus, and let's go a little bit to the overall goals of our project. So our idea and our intention, but also our acts, is to collect more than 400 tons of plastic, prevent 60 tons of plastic to end up in the ocean, organize 120 cleanups, and that's organized around the 20 tons challenge, a common challenge that is shared between different citizens, NGO groups, and organizations, and develop 12 innovative solutions that they have to do with the reduction of plastic in our oceans. Here there is a QR code that can link you to our website that is presenting like the different innovations of Remedies in the form of a marketplace. So if you are looking or if your projects or your stakeholders, they are looking either to scale, to invest, or to adopt like an innovative solution for a zero waste future, this is your place to be. So there is also like a dedicated page that is related to funding opportunities that they are also available, either from the Mission Ocean, but also from other sources that they could be private investors, banking, or foundation. You can find also like different details there that they are added related to the different eligibility criteria, the sectors or the respective regions. Okay, let's move to the core, that is like the remedies innovations. One of our first innovations is the aerial drones. It's been developed by our partners in Morocco. It's the Mohammed Premier University. What is it? It's an AI-powered drone solution that enables rapid practice environmental monitoring across different areas. So what we are doing there, and this is started already from the beginning of the project, is that we can having a deep learning system that is combined with drone operators, simple technology that it can be used from different stakeholders. This machine is combined together with acts of cleanups or research monitoring cleanups. So the machine is learning and it can identify pollution hotspots. It can also spot like waste or litter that is like above 2 centimeters, and it has like a very big accuracy already. Like from the beginning of the project, it was like ranging from 70 to 80, and now this has been improved as soon as more cleaners and more trials of the solution is being applied in the different regions. A second innovation is related to the dashboard. Here we have CBOS, that is the lead partner for this. It's a digital platform integrating environmental data from various monitoring sources into one unified interactive system. So this is somehow, let's say, the baseline of our different solutions. So we combine data that they are related to monitoring, beach cleanups data, but also BOIA monitoring system that we'll explain a little bit later, into one unified dashboard. So you can have like a check to evaluate the project's impact, or you can see like for a certain region, what is being applied, what are the results, and also take decisions and inform your strategy related to scalability or replication of a different solution that you apply in your region. Moving on, here we have like the microplastic filtration system by Clara Juan. This is a startup that is based in Slovenia, and they have applied the solution in copper. This decentralized water treatment system that is designed in order to capture microplastics and other contaminants at the source. So is a unit that it can be applied to ports. Like this is what we have been testing in copper. So it's capable of treating 1 to 5 cubic meters per hour, and it can reduce the pollution in certain spots, like hotspots of pollution. The unit has been supplied with IoT solutions as well as sensors, so we can have real-time monitoring of the solution. It's something that has been working and developed outside of the program, but also it got evolved within remedies. Moving on, this is Naftilog. This is also like an innovation that it was developed as part of Remedies. It's been developed and applied from MCD in Mykonos in Greece. So what it is a GPS-enabled buoy system that tracks the movement of plastic waste in rivers and marine environments. So what was this about? We managed to release like more than 100 devices, such devices, in river streams, but also into the Aegean, and we managed to trace the mobility of these devices in relation to water streams and weather conditions. The device transmits location data every 12 hours. And the idea is to map the pollution pathways and understand also concentration points. This was also like a solution that it was developed in collaboration with stakeholders, because like the devices can be found from someone and it has specific guidelines of how you can return these buoys back in order for them to start again their journey. Moving on, like this is a solution that is developed by CDDani, is a big company that provides fishing equipment based in Italy, and with a lot of different partners, of course, that they were like Next Technology, and also CNR, BioMe, AITIP, and University of Maribor. So this is also a big innovation, is a sustainable alternative to traditional plastic fishing nets, that they are fully degrade under industrial composition conditions. So it's biodegradable nets, as we can say. These nets, they maintain the strength and the durability and the usability, but they have an end-of-life solution that is aligned with the circular economy principles. These nets, they have been tested with fishermen also in Italy and in Mykonos in order to be able to get valuable feedback for possible adoption of this type of nets from the local communities. The idea is that they prevent the pollution and the microplastic generation as well, while they support fisheries in meeting EU sustainability regulations. Short-line marine litter application, version 10. We have a lot of versions, as you can see. So Infordata from Italy is the owner of, is the developer of the solution, along with Venice Lagoon Plastic Free and the University of Maribor. So it's a citizen science tool for standardized and transparent beach litter data collection. This is also what we identified as an opportunity within Remedies, that there are a lot of teams and organizations and citizens groups and activistic groups that they are doing cleanups, but we consider how we can support those, how we can elevate these actions by the right use of technology and protocols. So it's in an app for that allows users to record the location, type, and quantity of litter that they found, and it automatically integrates the data into a management portal. This is a more easy way for the citizens group to facilitate information, but also to see collective results in a certain region or to see evolution of a certain spot through time. Moving on, this is Wasser 3.0, PX. Is a company that is based in Germany. So it's an innovative solution for the detection of microplastics in various waters and their removal using agglomeration and fixation, including a full reuse concept. So this is a solution that it got installed in Mykonos wastewater treatment plant. Is an island in Greece. So it was a collaboration there in order to detect and to remove the microplastic from the wastewater treatment plant. It combines, as you can see here, of a physical and chemical process in order to be able to cluster the microplastic, is what you can see in the globe. And it achieves up to 75% lower operational costs while enabling full waste reuse, promoting sustainable water management. So the water that goes as an output, as an excess out of the treatment of the Wasser-free technology, is water that can be used also for irrigation or it can be used like for other purposes in agriculture or secondary uses. Another solution is the zero waste seaweed coating for cosmetics. We go more into prevention here. Nick from Slovenia is the inspirator and the creator of this solution. So it's a seaweed-based coating for cosmetics, for cosmetic products that eliminates the need for plastic packaging. Here you can see a workshop that it was created along with citizens. So you have like the product that is dehydrated, and there is a fine film of seaweed coating that is around it. So you have like single-use portions of shampoos or shower gels or other types of cosmetics. Yes, like this is a solution that it can be applied in hotels or households and supports the zero waste production. Moving on. This is a solution again developed in Mykonos. Is a mobile energy efficient water generator, H2O Oasis. The idea is to provide fresh water and energy efficient water and transform it into pure drinkable water for central points of the island. And the water is being produced from air moisture. So it has a photovoltaic powered system, and it's a concentration unit that can transform the atmospheric humidity into clean water. This can reduce the big challenge that we face in many islands across the Mediterranean, that is related to not potable water from the network water. So all the tourists and the visitors and the local, they have to drink from plastic bottles. The river cleaning system, the owner is River Cleaning from Italy, and the partner implementator is University of Maribor. It's a modular autonomous river system designed to intercept the floating plastic waste. It's been installed as a solution because we have studied and we know that rivers is the access point for the litter in the oceans. So it's fixed units that they rotate and they capture litter without having to disrupt the water flow. It's naturally flowed from the powered from the power of the river, and there is no need of external energy use. Okay, two more to go. Underwater water drone technology from Clara One, but also from Mohammed Premier University in Fortata and CNR. So it's a next generation underwater drone solution that can detect both micro and micro and macro plastics. So it's also an AI based algorithm that is combined together with sensors and sampling system, and it can automate, let's say, the litter monitoring in different zones and different coastal areas. And last but not least is the plug-and-play 2 zero-waste solution. So here is a solution that is developed by Alchimia Nova Greece, BioMe, and CBOS, AITP, and TUA. So it's a closed-loop a reuse system that is designed in order to have seamless, as we say, event integration and on-site cleaning infrastructure for reusable cups and for reusable cups in festivals or bigger gatherings. So this is like a big container. What you can see here, it's like a big laundry machine for cups cleaning, like on the spot. So this can reduce up to 70% the plastic waste that is being created in big festivals or conventions. So it's a decentralized solution, it's a movable solution. Sorry. Is a movable solution, and it can be working. It can be applied in big conventions where you can use more than 2,000 items be washed per hour. I think this is it. This is our partners. As I said before, the marketplace is available on our website. So you can directly contact the creators or the innovation holders, or also us from the Impact Hub or the NIC, that is the coordinator of the program, in order to get more details regarding the solutions and the innovations applied. Thank you.
Thank you very much, Dimitris. Very interesting. And we will move to IMermaid project and Jasmine. So Jasmine, you can take the control and move to your slides.
Yes, thank you. Let me see if you can see my laser moving.
Yes, yes.
Yeah, okay. Thank you. Hi, good morning. I'm Jasmine Cecconi from SMAT, which is the Integrated Water Service Manager of the metropolitan area of Turin, which is located in the north part of Italy. And today I will present you one of the use case of the IMermaid project. IMermaid project is an Horizon Europe funded project, which is mainly focused in the development of innovative solutions for water monitoring and treatment to protect and decontaminate the Mediterranean sea basins from the contaminants of emerging concern. This is a project that is almost ended, since it will end at the end of the next month and has a duration of 36 months. One of the main objectives of the project is to develop and test advanced and innovative solutions to prevent, monitor, and decontaminate the Mediterranean Sea basins from the contaminant of emerging concern. We have also other objectives, for example, to raise awareness to the citizens about how to handle and dispose the chemical compounds. In the right part of the slide, you can see all the 5 use cases that we performed on IMermaid. We can start for the first one, which is located in a buoy in Cyprus. The second one, which is located in Italy, in a wastewater treatment plant facility that I will present today. Then we have the third one, which is located in Spain, in another wastewater treatment plant. Another one in Tunisia, in a wastewater treatment plant facility located in an industrial site, in a pharmaceutical industry. And the last one, which is located in Greece, in a landfill for the monitoring of leachate. As I was mentioning, I will present today the use case number 3, which is entitled Pilot Plant for the Removal and Monitoring of Heavy Metals from Wastewater, which is located in one of the wastewater treatment plants that is managed by SMAT, that is located in Brandizzo, near Turin. This wastewater treatment plant treats both industrial and domestic wastewater that, after being treated, are released in the nearby river. The problem that we wanted to target with this pilot is the fact that if high concentration of pollutants reach a wastewater treatment plant, these pollutants can be not completely removed by the plant, and they can be released in the water body. So the pilot to assess this problem had a duration of 2 months in last April and May 2025, and was focused in the demonstration of mainly two technologies. The first one, which is developed by the Finnish startup Wefiner, called the Fordy Scavenger, for the removal of copper, zinc, and aluminum, and the second one developed by the University of Florence, which is a heavy metal box based on an electrochemical sensor for the monitoring of zinc, cadmium, lead, and copper. Here I have reported a very detailed timeline of all the activities that we performed, starting from the preparation to the site until the end of the pilot with the waste characterization and disposal. I will present you the main points. First of all, we had to define the best site where to install the technologies. That came out to be Brandizzo wastewater treatment plant. You can see here an overview of the plant and of the treatment steps. And then we have to define also the best installation point of the technologies. We did that performing some tests and also shipping samples to the technology provider. In particular, we take into account two possible points of installation. A first one, which is located in the fine screening step, so at the beginning of the treatment that wastewater undergoes, and the second one, which is at the end of the treatment process in the final disinfection tank. To define the best installation point, we had to take into account the requirements provided by the technology provider. In particular, for the removal technology, the TSS content in the wastewater should be below 5 ppm, while for the monitoring heavy metal box, we had to obtain a TSS content below 25 ppm and a BioD below 15 ppm. What we did to define the best point of installation is to consider the historical trend of these parameters in the two investigated possible installation points, and you can see reported here the data for the final disinfection tank. We also had to take into consideration the fact that Wefiner, the supplier of the Fordy Scavenger technologies, was able to implement and develop a filtration system that were able to remove TSS content sufficiently if the content of TSS was below 20 ppm. So looking to the historical series, we can see a medium value of TSS content that is about 7 ppm, so we can conclude that the best point of installation is in the final disinfection tank. So in April 2025, we finally were able to host both the technologies. The Fordy Scavenger were shipped from Finland inside a container. So we used a crane to place the container and the technology in the wastewater treatment plant. We had to provide electrical connection, well water. We had to supply reagents and equipment and prepare some solution that were needed for the metal box that you can see in the photo for to perform calibration and maintenance activities. We also have to provide on-site assistance for the maintenance activities and to optimize the analytical protocol of these technologies. During the pilot, we also collect some samples in order to validate both the technologies through analysis that were performed in our accredited laboratory. Of course, during the pilot, we had to address some challenges for both the technologies. We can start with the heavy metal box. I'm sorry that the slides, in the slides there was some movement, so we cannot see very well the titles. What we experienced was a high electrical noise in the instrumentation that was due to the other instrumentation that was connected in the wastewater treatment plant. And we solved these problems changing the pretreatment protocol of the sample using hydrochloric acid instead of acetate buffer, grounding the instrumentation and using a noise filter. Then we also had the formation of some air bubble inside the measuring cell that prevented us to perform the measurement. So we had to purchase an additional tank and an additional peristaltic pump in order to pump the water inside the tank, let the water set there in order to the gas for 2 hours before analyzing it through the heavy metal box. In this way, in these 2 hours, the dissolved oxygen were able to be released in the atmosphere, and in this way we remove the air bubble formation. Through this approach, using hydrochloric acid, we were able to perform analysis for cadmium, lead, and copper with high sensitivity. You can see here a real voltammogram of one of the samples in Brandizzo with a signal that is connected to copper. But this approach wasn't able to give us a sufficient sensitivity for zinc analysis. For these heavy metals, we have to switch back to the initial protocol of pretreatment of the sample using acetate buffer, and in this way we were able to obtain very good voltammogram in real samples of Brandizzo. So you can see here the signal related to zinc. Regarding the Furnace scavenger, so the technologies for the removal of heavy metals, in this case, more regeneration cycles than foreseen have been necessary, because once the capacity of the filters to remove the heavy metals was full, we have to use two reagents, one acid that you can see here, sulfuric acid, and a base, to wash and regenerate the capacity of these filters. During the pilot, we experienced that more regeneration cycles were needed than foreseen. So briefly, you can see here the results reported for the metal box. In the left part, as an example for copper, you can see in blue the data that were provided by SMAT laboratory to validate the data provided by the heavy metal box, and you can see how these data are very similar. So we can conclude that the heavy metals were able to provide us reliable results and to monitor heavy metals at concentration level that were at least one order of magnitude below the low limit for the discharge of wastewater into surface water. You can see here the low limit, which is 100 ppb, and we were able to monitor about 10 ppb, so one order of magnitude below the low limit. What was needed at this point at the end of the pilot is to reach the KPI and to lower even more the limit of detection of cadmium. So more analysis and more tests needed to be performed at the University of Florence laboratory. Regarding the Furnace scavenger technology, you can see in the right part a graph when the dotted line are all the regeneration cycles that were needed, that you can see were a lot and more than we were expecting. So also for these technologies, after the pilot, additional tests needed to be performed. I will explain very briefly the post-pilot activities that were done in order to reach the two KPI. The first one for the heavy metal box is to reach some limit of detection that were below the predictable no effect level concentration that you can see reported here. As you can see, during the pilot, just for cadmium, we were not able to reach a limit of detection that was enough low. So we had to do some additional tests at the University of Florence, and they did some adjustment in the protocol of measurement of the system, and you can see how we were able to lower this limit of detection below the P and EC value, and in this way we were able to achieve the KPI for all the targeted heavy metals. Regarding the Furnace scavenger, the KPI was to reach a removal at least of 95% of the heavy metals from wastewater, and also in this case some post-pilot activities were performed by the technology provider. In particular, were performed two kinds of tests that were performed in double. The first one, which is an off-site pilot using a synthetic feed solution, so tap water that which were added some heavy metals, and you can see from the graph how until 1500 they were able to reach the removal efficiency that were sufficiently high for all the three targeted heavy metal, copper, zinc, and aluminum. And then they also did another pilot on site in a real facility, an industrial facility in Finland, just for zinc, and for bed volumes below 500 or 1000, they were able to reach this removal efficiency. So also for these technologies with post-pilot activities, the KPI was reached. That's all for my side, and thank you for your attention, and let me know if you have any questions and we reply to them.
Thank you very much, Jasmine. I also don't forget that on the chat I post a link that you can drop all your questions, so at the end we can answer them. And not to waste our time, Angel is the next one from Remediation Project. So Angel, the floor is yours. You can take control and start presenting.
Thank you very much, and good morning, everybody. My name is Angel Muñiz from the European Marine Board Secretariat, and I'm here to represent the Remediation project, which is actually a sister project of the Armormate. So it was really interesting to see how they solve many of the challenges that also somehow this remediation also face. So that was also really interesting. I have the honor, but also the responsibility to try to summarize the remediation project in 10 minutes because this project will finish in 1 month. But basically, remediations is to reduce chemical pollution through innovative nature-based water treatments. Kind of like a narrative, and I'm taking advantage of the footer of the presentation template. So this is a mission project. So the EU missions to restore our ocean and waters is an initiative of the European Commission to advance technology development, innovation to actually solve solutions the society faces, like for instance the degradation of our marine environment. One of the objectives of this mission is to prevent and eliminate pollution of our seas, oceans, and waters. And then to implement the mission, we have what we call the mission lighthouses. So in each sea basin and also in the Danube River, there is a target objective. And for instance, in the Mediterranean is to prevent and eliminate pollution. As you might know, in the Mediterranean, humanity has been living there for millennia. So we live there, we grow crops there, we fish there, we also use it for transportation. And then in more recent times, we also do heavy industry. We might have like a more industrial developments. And also that means that it's actually being polluted. So that's one of the things that remediation is a Mediterranean lighthouse project, and also to aim to distress the Mediterranean from pollution. Just as a little explanation about wastewater treatments. So of course, when you have water for instance, from a city, you have to filter the water when it comes into the water treatment plant. Then you have the primary treatment, where it's by basically sinks the particle matter that is in the water. Then you have a secondary when you have like a microbial breakthrough of organic matter, and then you can have tertiary, which is also treating nutrients, and then you can have also a quaternary if you want to target more specific pollutants like pharmaceuticals or another industrial waste. And then also you have disinfection to remove potential harmful organisms. Just as a reference, this is for environmental protection regulations. This is a black box. So in general principle, the pollutants that have to go in has to be bigger than what comes out. So in principle, whatever goes in and whatever goes out has to be cleaner. So in general, regulations doesn't care what kind of technologies used, what kind of processes, has to be cleaner afterwards. So that's also the added value of the remediation system, that also can be plugged after any of the already existing treatments. So you can see here a picture of the system when you have some containers and also the photobioreactors. And the added value of this system is that it's macroalgae based. So it's basically the macroalgae present in the water will do the dirty work, let's say, to clean the water. On how does it work? And also I will come back to that we call it the remediation approach. So you have the water that you want to treat. It could be natural waters, but also it could be from wastewater. Then it goes to the system. As I said, with the container and the photobioreactors, and then within this container, you have at the back, you have the dirty water that also been, with a different probes and different measurements, because of course you need to assess what kind of pollutants are in this water, and you know what needs to be treated. So you need to measure this. Then that water that has been measured, it goes through the different photobioreactors. So basically, these photobioreactors, they promote the growth of the algae that is naturally present in the water. So you don't have to add any other species, you don't have to add any chemicals and so on. It grows naturally. So if you are a particle of water, you will travel through these different photobioreactors until you reach the end, and of course you can determine how long will the water stay in this system or not. So that's also quite flexible, depending if you have a really dirty water or many nutrients to pollute and so on. So of course the macroalgae will use the nutrients that is present in the water, and then also they will capture different pollutants that is present in the water. And it's also these photobioreactors. They act as a kind of like an ecosystem in its own, because then when the algae die off, it will get remineralized by the other algae, use a nutrient, so it's you don't have to clean it up too much. Then this water goes to this other tank that you can see at the forefront. You can see that the water is a little bit clearer, and it's also something that I learned, because I'm not really expert on wastewater treatment, is that of course you also have the color of the water. So if you see that is the color is brown, it means that, you know, it doesn't mean that the water is dirty, it's just that the color of the water. So then, of course, they measure it again to see how the pollutants has been reduced and so on. And then, of course, we tested this technology, and then it wasn't released to the environment, but we also have a system where you can monitor, and you can see one of the buoys here in one of the test cases, where it was monitored that the pollutants in the environment and so on. So this is just a summary infographic of what I just explained, and it's also I will present in a bit the different test sites. But then I want to highlight here is that the information coming from the sea trials. So basically, all these buoys that are in the environment at the end of the rivers, or also in marine waters. That information is transmitted to the European Maritime Observing and Data Network, so EMODNet, which also could be, contributing to the digital twin of the ocean. The testing sites, and it's also I will see, and I will put different images of how the evolution of the water. So one is in Taranto in Italy, where it was also the natural water. So this is when they just installed and you have like clear water. Then we also tested in a wastewater treatment plant in Attica in Greece, and then also in wastewater treatment in Kocaeli in Turkey. We helped local authorities in Bulgaria, Egypt, Moldova, and Morocco to have replication strategies and roadmaps to potentially implement this kind of systems. So of course we didn't fund the implementation of this technology, but just to check, what kind of water, what kind of objectives do they want to clean the water and so on. So this is also in terms of for other projects, this is what we did, where a call for associated regions. Also, what I mentioned is we call it the remediation approach, because of course, not only the technology and the system, but also there was an important component is stakeholder engagement. So we did at the beginning in the different test sites, and also we will do in the next month other stakeholder workshops, and also we engage with the local communities and local policymakers, and also they had visits from schools and so on. So in general, people see it as a positive social impact, because as the macroalgae is doing the dirty work, you don't have to ask for exotic chemicals and so on. So it could be also locally produced, the things that you need. So it also could be a turn and a positive turnover to the local communities. Just to present the results and recommendations, and I'm happy to announce that the A policy brief explaining all this is now officially launched, so you can scan the QR code, and in a bit I will put in the chat the link to download it. So as a result, the remediation system is scalable, adaptable, and also can accommodate local and seasonal variations. You can have also more information in the policy brief on what kind of authorities are targeted for this kind of technologies. We proved that has been removal of nutrients, metals, industrial chemicals such as PFAS, pesticides, and pharmaceuticals from wastewater and natural waters. And also there is a reduction of operational expenditure. As I said, the macroalgae do the dirty work, and also has much lower carbon footprint and facilitated treatment of urban industrial wastewater for reuse, because also in the Mediterranean, every drop of water is important. So it's also really important that the reuse of this water, of the treated water, is promoted. In terms of recommendations, of course, the most important one, that is pollution prevention must be a priority, because we will be never able to treat 100% of the pollution. So it's cheaper to treat the pollution that is not there. So it's better to to prevent pollution entering in the Mediterranean or other environments. Of course, we also promote or call for establishing clear and specific removal targets and standards for these emission levels of emerging pollutants, because as you might not know, but then for PFAS, there are currently like 10,000 different sorts of PFAS, so we have to know which one do we want to target, because to measure, because it's impossible to measure all and then also to treat all. Also for the biomass that is potentially coming from these wastewater treatments. We also call to develop a standardized monitoring and a clear technical guidelines, because sometimes it depends on how much the water, if it's going to be used for drinking, if it's going to be for reuse, if it's going to be used for agricultural purposes and so on. You have different ways of measuring and so on. So there should be a standardized monitoring and guidance on that. And also in the revised wastewater treatment directive that is currently in place now at the EU, this wastewater treatment needs to reduce the CO2 emissions. So there is also should be some standards on how we're going to do that and so on. Of course, we call to recognize microalgae-based wastewater treatments, because this is not really currently used in wastewater. So this is a nature-based solution. And then also we call for to incentivize to promote the deployment of real-time monitoring and early warning systems, because if you have this kind of buoys in natural environments, you can also detect accidental leakage from industry and so on, so that could also provide early warning and not you don't have to wait to the environmental impacts. And then lastly, we call to incentivize the public-private academic partnerships in diverse sectors, because for instance, these kind of technologies could be used for wastewater treatment, so to treat wastewater from hospitals, for instance, because you will have a lot of pharmaceuticals reaching the environment, so it could be used for that. And then, of course, the kind of collaborations between public and private organizations can also explore alternative uses of the resultant water and also biomass, which also could be used for energy generation. Finally, I would like to give all the full credit to the team, to the consortium, and this is a group picture that we take in front of the one of the test sites. So of course, I'm just presenting. I'm just a messenger of this project, but I'm happy to be part of this project. It has been really interesting. I hope you enjoy the policy brief. You can always ask me some questions, so I will leave it like that. So thank you very much for giving me the opportunity to present the remediation project.
Thank you very much, Angel. Very interesting and very comprehensive presentation. We have 2 questions on the chat, but we will ask them at the end. We will answer them at the end. I also put it on the slide that I shared. And because now we don't have so much time, that's why I prefer that. And we move to mobiles project and Angelo. Angelo, you can take control and start your presentation.
Okay, thank you, Krisa. Good morning, everyone, also from my side. So the title of my presentation today that represents mobile and part of the coordination team is using aptamers as sensing elements in electrochemical sensors that are called also apta sensors. The presentation is divided basically in 4 parts. The first 2 is just an introduction about the project and then about electrochemical sensors in general, and then I'll tell you some details in what we are developing here at the National Technical University of Athens. So the full project title is Monitoring and Detection of Biotic and Abiotic Pollutants by Electronics, Plants and Microorganisms-based sensors. It started roughly 20 months ago, the project, and it will end at February 2028. Here you can see all our communication channels that I invite you to visit. And about the partners, we are 15 participants from 9 European countries, plus 1 associated that is from Israel. The universities are the majority. The NTUA is the coordinator, as I said, but we have a university from Spain, Italy, Germany, Serbia, Cyprus, and France. We also have some research centers from Europe, again, Italy. This is the research from Israel. It's called the Volcani Institute, France. Then there is 2 partners from Poland. One is the Institute of Soil Science, and one is the Pro Academy Research Center, and 3 SMEs, France, Germany, and then Czechia. So, as I said, I'll give you just very brief information about the project, then I will focus in one of the topic. So we have 3 pillars, basically. One that we named it detection, where we're using electrochemical biosensors. We will detect pesticides, hormone, and this is what I'm going to talk about, the biosensor for estradiol. And then we can detect also pathogens, antimicrobial resistant bacteria, and also genes. Then there is the pillar of monitoring, where we use living organisms, basically plants, bacteria, and the diatoms to detect all these kind of pollutants. Just to give you an example, at Rome, the University of Rome is now manipulating the genome of Arabidopsis thaliana in a way that when arsenic is present in the soil or in the water that is used to grow this plant, the plant will become red instead of green. So this is something that can be used for passive monitoring and gives you information if a soil contains or not chemicals. We developed for arsenic, but of course, by changing some genetic elements in the plant, they can detect also other materials. And then the third pillar, that it's about the metagenomic analysis of soil microbiome. So from 5 different sites in Europe, we are collecting polluted soil where we will analyze it using genomic tools. So we will sequence the DNA and the RNA that is extracted from the soils in order to identify what are the microorganisms that live there. For 2 reasons, basically. One, to understand how the microbes are affected by pollution. And second, to see if there are some kind of markers or some kind of pathway that we can take advantage and then maybe use to dispose, to eliminate the pollution that contaminated the specific soil. But as I told you, today we will focus just on a little part of electrochemical biosensors. So what are these electrochemical biosensors? Basically, they are formed by two components. One is the bioreceptor, that is also called the biorecognition element, and then there is an electrochemical transducer, so an electric circuit. Here you can see these two elements. So we have we can have antibodies, piece of DNA that are called aptamers, enzymes, whatever, and usually they are attached, they are immobilized on the transducers. That is the electrode part of the system. So when these two components are together, they form a sort of stable system where when we have chemicals, so the target pollutant or any kind of analyte that touch the biorecognition element, it triggers a sort of electric signal that can be detected. And there are different techniques that we can use. This differential potential voltammetry, cyclic voltammetry, square wave voltammetry, electrochemical impedance spectroscopy that basically we use, and each one of these techniques gives a sort of wave, and the wave is perturbated when there is the binding between the biorecognition element and the target pollution. Now, the typology of electrochemical biosensors are a lot. Usually, we use natural bioreceptor that can be found in organisms, and this, as I said, they are antibodies, enzymes, nucleic acid, but also virus. These are bacteriophages are virus, whole cells, membranes, or also organelles. And they, let's say, are a small part of a more general family of electrochemical sensors. So here we have, when you use a bioreceptor, we call it biosensor, but you can even use inorganic stuff like nanoparticles or organic linkers, both synthetic or just natural. So you can construct with basically almost any kind of material a sensor where you immobilize a receptor on an electrode, and then you measure how it changes the properties of the electrical properties of the electrode. Now, the problem working with natural bioreceptor is that they are unstable. So using antibodies, enzymes, even cells, they require physiological conditions that are very difficult to maintain on an electrode surface. So what we do, usually we use bio-inspired molecules and the aptamers, like nanobodies or peptides that can be synthesized in the lab without any living cells or without any animal. They are the best molecules because, as I said, you can do the synthesis in the lab and then try to immobilize them. And these molecules are also known as molecular imprinted polymers that can be easily produced and are available in the market. Now, let's focus on the aptamers and what are they? So they are single-strand DNA, RNA, or XNA, nucleic acid. I will come back on this X. And they fold in a 3D structure. And this 3D structure gives them the ability to bind. Here is a biomarker, but can be pollution, can be anything. And this binding, it's highly specific. So when the biomarker attaches the target aptamer, this means that we have high specificity and high sensitivity. Just to tell you what is the X here, usually you can have synthetic nucleic acid where the sugar, the R and the D, basically it's a ribose and the deoxyribose. There are two molecules that are sugar. When the sugar is substituted with other molecule, you can have what is called XNA. But basically, they they give you a polymer of nucleic acid, and it's a standard technology. I want to also to show you how easy theoretically it is to produce or to select an aptamer. And that's why we use them, because they are easy to be produced. And the technology is called systematic evolution of ligand by exponential enrichment. That is, in short, SELEX. So once you have your target molecule, you can, let's say, put them in contact with a library of aptamers that are a random sequence of DNA, but not exactly random. In any case, they have to form this 3D structure. So you put them in contact, you allow them to react, so they form some complexes. And once they form the complex, you isolate the complex, and then you extract just the piece of DNA or RNA, and you amplify it using PCR. That it's a standard technology that is used in molecular biology to amplify the DNA. And you have your aptamer that is enriched. So if you repeat a couple of times this SELEX process, you end up with a very good aptamer that has a high affinity and high specificity for the target, and it's comparable with the performance of an antibody. So in the market are already available a lot of aptamers for a variety of targets, both pollution and non-pollution. But in case there are companies specialized where you can ask just an aptamer, and they, in a matter of days, can produce it. Now, I'm going to show you the apta sensor that we are going to use for the estradiol. And just to help me, I will do a sort of recap. So we say that for an estradiol biosensor, we need basically two components: the biorecognition element, that it's the aptamer plus the transducer. Then we need to immobilize the aptamer on the electrode, and then measurements. So we will see now point by point these four steps. The first thing that we use is the electrode. These are called screen printed electrode, and it's gold. Here, this yellow material, it's gold. It's very few thin layer of gold. And then there is also a third one that it's silver, but this is just how it works, because we need to have some reference electrodes. So usually we use this standard one. Now we also test others, and I'll show you later. And then you need what is called the transducer or the readout circuit. That it's an electric motherboard. We customized them, we designed, and we bought all the components in order to make it. And finally, you need also the aptamer. So these are the aptamers that we are currently screening, because we want to be sure, we want to isolate one that has very high sensitivity. We took them from the literature, but we modified them a little bit. And the most important modification is to attach this thiol group at the 5' and at the 3' end, not in the same batch, but in separate batch. Because this thiol group is the key element that allows us to put the aptamers on the electrode. And here is what we use basically to attach our aptamer to the electrode. We use a sort of characteristic covalent bond that is formed between gold and sulfur, and this gives a sort of very robust but modifiable interaction that it's of paramount importance in stabilizing nanostructure and transmitting electronic interaction between gold and sulfur-containing unguarded molecules. What does it mean? So this is, let's say, imagine at atomic level the gold surface. The orange dots is the sulfur group. And then this hexagonal shape can be anything. So it's a radical group that usually is indicated like this. When we leave the sulfur group, in contact with gold, they spontaneously bind each other. So they form a sort of very strong covalent bond. And whatever is attached to the sulfur group also remains attached permanently and covalently to the gold surface. But this gives us the possibility also to see electrical change, because if the aptamer that we attach to the gold surface has some kind of perturbation when it binds to the target molecule, we can detect it because it's transmitted through this covalent bond that is formed between the sulfur and the aptamer. Of course, the sulfur group is attached also covalently with the aptamer. And another good properties is that when you have something that is tagged with the sulfur group, it spontaneously forms the self-assembled monolayer. So you are sure that you have just one unique and homogeneous coating of aptamers, and they don't attach one upon the other, so they just stick on the gold surface in a very smooth and clear way. This properties is maintained also if you use gold nanoparticles. So this is a gold nanoparticle, for example, can be also used to form a sort of polymer using the gold atoms as a linker. And just for the records, the sulfur also binds copper and silver, but copper and silver usually they have problem with oxidation, so we prefer to use gold. So now how we are doing our electrochemical biosensor. First of all, we just order the primer, the aptamer, that is tagged with sulfur group, and we deposit just 30, 40 microliters on the center of the electrode. And here there is the stem loop structure of the aptamer that binds after a few hours, thanks to the tile group. And after we connect our electrode to the rest of the readout circuit, and all the system now it's ready to measure the estradiol. And when we apply estradiol, we can see that based on the quantity of the estradiol that we have on the solution, we have different amplitude of the curve. And we now are testing it for the sensitivity, and we, of course, it's not finished, the system, because we need to put a case on that. We need also to put a display, because now it works with a laptop, with a computer. But we, at least now, we know what is the best primer, what is the best aptamer. Sorry, sometimes I say primer, to use to identify the estradiol. We also worked with another kind of electrode. This is called a capacitor, but it's very, very sensitive. But I want to show you this video because besides the, let's say, electric system and the bioreceptor, we have also to optimize a way to dispense on top of the electrode an accurate volume, a standard volume, because the more you put, the more it reads. So in order to have a sort of quantification, accurate quantification, we had to be sure that every time we dispense on the electrode a very precise amount of liquid. And we Edentech, that it's a partner of the project, developed this microfluidic device where independently by the amount that you load on the inlet channel, it dispenses just 30 microliter in the center of the system. So this now will be improved and will be adapted to the new design. And we will then have a microfluidic device also for the new system. Here, it's another way that we can measure the presence of estradiol. In this case, here we used capacitance measurements. So this is the curve with the aptamer, but with no estradiol. When we put estradiol, we notice a sort of drop on these electrical properties. Then if we wash it, the drop still remains, but there is a lot of noise. And that's why we prefer to change the electrode. And this is, for example, the same experiment, but instead of using the aptamer that recognizes the estradiol, we used a random piece of DNA, so we don't have any measurements. And I think this was my last one. Yes, thank you. And for me, it's all.
Thank you very much, Angelo. Also, don't forget, if you have any questions, to drop them at the link that I sent you previously. So we will move to the next presentation to 0 PM project and Sarah. And after that, we will have a 10 minutes break just to have a small break. So Sarah, take the control and start your presentation.
Great, so I have the control, but do you see it in the big view?
Yes, we can see it.
Yeah, okay, I see a different view. Okay, great. So I'm going to be presenting 0 PM, which is a project which is also actually nearly finished. It finishes in September this year. And we interlink 3 different strategies: prevention, prioritization, and removal strategies to protect human health and the environment from persistent, mobile, and also toxic substances. So we have a project that's very heavily inspired by the chemical strategy for sustainability. We use the same kind of triangle, the same color scheme, but we also take the view that we need to interlink all these 3 different strategies. Otherwise, we won't be able to clean up the environment and improve our livelihoods and reduce costs of basically everything. So what we do in 0 PM is we have 15 different partners working together across these work packages. We have 3 different work packages that focus on prevention, 2 that focus on prioritization, and 1 that focuses on removal. So today I'm going to talk about the work package that focuses on removal, since this is what the webinar is about. And what I've decided is, first of all, I'm going to go through each of the different these are actually different slides to the ones I sent, but never mind, we'll just use these slides for now. I will tell you a little bit about each of the work packages and then talk about the results that we have. So what we try to do in our prevention work is we're looking at alternatives assessment, policy, and market transition. So we are trying to find different substances to these harmful, persistent, mobile, toxic substances, and work out ways of using them by different stakeholder groups to reduce exposure and minimize risk. We're also looking at policy. So at the moment, there's lots of movement around specifically PFAS policy. So we're trying to find ways that we can feed our results into different policies. And we're also producing some policy briefs at the moment. So there'll be about 11 policy briefs coming from 0 PM that you'll be able to have a look at. And we work a lot with different companies. So some companies are able to transition away from these substances. Some companies are at the start of this transition, I would say. They don't necessarily have the knowledge and the tools. So we're trying to provide them with different things that they can actually use. One of the things that we've made, I'll explain in a second, is the PFAS guide, where companies can go and click and see whether they have PFAS in their supply chains or not. What we do in the work packages related to prevention and removal is we're looking at how we can group substances. So the universe of PMT and VPVM substances is huge, so we need to try and find methods to group them. A good example is PFAS. PFAS fall in this group, and this is now how they're going to be regulated. But there's also other ways that we could group them by looking at things like dead-end transformation products and put those as a group. They all start from different things, but they end up as the same thing in the environment. And TFA is an example here. We have a work package that looks at risk assessment. So we're trying to find different and better ways to do risk assessments, specifically promoting the inclusion of NAMs in policy. I think this is one of the things that lots of projects are working on at the moment. And we're also looking at technical solutions. So for our technical solutions, what we're trying to do is work with water and also sewage sludge. These are the two media that we've decided to focus on, and we're looking at innovative solutions for how we can remediate and remove these persistent mobile toxic substances. What we have done in 0PM is we focused on 3 different groups. So when we first started, there was some focus on PFAS, perhaps not as much as there is now, but we're looking into PFAS, and we're looking into triazines, and we're looking into triazoles. So these are groups that within the group themselves, there's some substances that are already regulated, and they all contain problematic moieties, so parts of the chemicals, which means that we know when they're in the environment, there'll be a problem. So what I wanted to focus on a little bit was to show you some of the results. I'm not going to click on anything, but when you get the slides afterwards, you can go through. So what we've done in 0PM is we've made an alternatives assessment database. So this is basically a large Excel, but turned into a database where you can go and find functional alternative chemicals to PFAS across different uses. So we took a use where we know that PFAS is being used. So let's say ski wax. Then we looked at why was PFAS being used in the ski wax. Then we looked at what alternatives could we have instead of the PFAS in the ski wax that would still deliver the same product function and the same level of function. So that's the database there. And at the moment it's just for PFAS, but we will extend it to different PMT substances. We also work with behavioural psychologists in 0PM to try and understand why people think certain things might be essential, or why people want to use products and what they think about having PFAS in their products. So we have a lot of data where we've spoken to different stakeholder groups to see how, for example, the European Commission could implement the essential use concept. This is very difficult because some people think essential, for some people essential means one thing, and for other people essential means a different thing. We're also working with improved life cycle assessment methods. So often these frameworks are not necessarily taking into consideration the properties of the PMT substances. So because they're persistent, they're in the environment for such a long time that it means that additional factors need to be considered. What we've done is also made a regulatory watch. This is a free tool that you can go to, and we update it every month to say what's happening within the framework, the policy framework of these substances. So we can track when the Commission is going to update the pharmaceuticals regulation, how far they've come in their discussion about the water policies, and it's just a really useful tool because it tells you what's going to come next. We've made a PFAS guide, so this is where companies can go and have information that I would say is more easily accessible to them, so they can see, do we have PFAS in our supply chain? Or perhaps we do, maybe we should focus, and then we give them some information about, what is PFAS, because it's not a given that everybody knows. And because supply chains span many countries, it's also quite difficult for countries with less knowledge perhaps to be at the same level as countries with more knowledge. We've also made a lot of lists of different ways to group substances. So in PubChem, we have a PFAS and fluorinated compounds tree. So it's a tree that contains millions of substances that are classified as PFAS according to the OECD definition. But then you can divide the list and you can look at, these are the substances that contain this many carbon fluorine bonds. These are the substances that are also on a different list in addition to the OECD list. So it's a really handy tool if you want to try and see how many PFAS could be classified in a certain way. We've also made an application where you can go and text mine, basically. If you have some text, you can look at transformation reactions. So to try and find out what would happen in the environment, if there's something written down in a publication, we can apply the tool that we've made and it will tell you what the dead-end transformation products are. And this has also been turned into an online tool where you can simply click on a different molecule and see what were its precursors, and maybe look at the different reaction pathways. That's very useful. And we've also looked at some in vitro hazard profiles for different substances, different compounds. And this tells us how hazardous they are in comparison to each other. And the idea with this data is that we combine it with exposure data to try and make a kind of prioritization or a ranking to say which of these substances should we focus on, which is the one that's most likely to have the highest risk. Those are just some ideas of the tools. And that was what I just wanted to say. Thank you very much, Sarah. Very interesting. If you have any questions, please drop it all in the chat or in the slide. Now we will have a 5-minute break, so we will be back at 1:30. So just take a coffee, relax. We have 3 more presentations and the Q&A session. See you in 5 minutes.
So we are back. Just a short coffee break. I hope you are here. And Vespina, can you hear us?
Yes, I'm here.
Okay, thank you very much. You can take the control and start your presentation.
Mm-hmm. Okay. Just a minute.
You can move the slides.
I cannot see the slides. They are not like they are still uploading for some reason. I don't
Okay,
know.
no. Okay, no.
Oh. But start from the beginning.
Yes,
I think
from the beginning. No worries, I will take the control. I will move to your
Okay,
slides
okay.
and I will give you again.
Great, thank you.
You can try now, take the control.
Yeah. For some reason, I cannot take the control. I mean, I pressed the button, but I cannot see the black window. Ah, okay. Yes.
Now?
Yes. Yes, great.
Okay, thank
see
you.
this? Oops.
Move to the next slide.
I think it started from the beginning.
No, no worries. I will take
I
the
don't know
control.
what's happening.
I will move the slides. When
Okay.
you want to change the slides, please.
Yeah.
Okay, you can start. And when you want to change the slide, please tell to me.
Okay, so I will not take the control.
Now you take it.
Okay. Do you see them? Ah, great. Okay.
Okay, perfect.
Did you see the slides?
Yes, yes, yes.
Okay, okay, great. So, hi everyone. I'm sorry for this for the delay. I am Despina Varamogianni Mamatsi. I am a postdoc working for a Bioscience project, and today I'm gonna share with you the overview of the on-site biological sensing for aquatic pollutants and biohazards. So, Aquabiosense project is a EU Horizon awarded project that is comprised from 8 partners across European countries and has a duration of 3 years, starting from 2024. So, our main aim is to make the tools for quantifying aquatic pollution accessible by everyone. And that's why we are developing easy to handle and portable devices in order to allow to customers to detect different types of aquatic pollution at a point of interest without having previous technical expertise. So by this means, we are aiming to make the water quality monitoring more efficient and affordable and faster by everyone, and allow users to have low cost and timely answers instead of waiting many hours for the lab results. Of course, this opens up the opportunity to being used by many kinds of customers, such as stakeholders, fish farmers, as well as industry operators, and not just scientific labs. So our systems are using high sensitivity analytical approaches based on antibodies, nucleic acids, or whole cells, in integration with state-of-the-art biosensing technologies such as acoustic wave transducers, fluorimetric systems, as well as organ-on-chip systems, as you can see in the figure. And real-time data can be interpreted by using simple and easy to handle smartphone apps. Here you can see the list of the target analytes described in Aquabiosense proposal. It covers a wide range of pollutants that can be found in aquatic ecosystems, such as hormones, antibiotics, or pesticides, naturally occurring toxins, microbes associated with harmful algal blooms or disease outbreaks, as well as heavy metals. For each target analyte category, we use a specific analytical approach, and we employ concentration ranges that are aligned with the European Water Framework Directives. For instance, by using the immunoacoustic sensing approach, we were able to detect pesticides such as bifenthrin or microcystin LR, which is a very toxic, which is a very harmful hepatotoxin. We could detect both pollutants at concentrations that were at approximately or even below the European regulatory limit. And low-cost results could be derived by using minimum water volumes and within 1 hour or less. So regarding the portable devices, we use for each pollutant type a specific setup. For instance, for organic contaminants, we have designed a pre-concentration system in order to elute the analyte into higher concentrations and smaller water volumes in order to be able to detect it with desired sensitivity in the immunoacoustic portable device. For the quantification of microbial RNA, we use cell capture and RNA extraction tools, and quantification is implemented using an optical fluorimetric system. Also, we use whole cell biosensors based on diatoms or fish gill epitheliums in order to determine heavy metals in aquatic systems. Last but not least, a sample procedure is being scheduled across many hotspots in Europe, such as Southampton Port or Dublin Bay in Ireland, where our developed portable devices will be tested with real or simulated samples. Since we are at the last year of our project, current work is focusing in finalizing the whole design of our sensors, and validation of these devices will be demonstrated by using simulated samples first in the lab and at a later stage at various places where pollution has been documented across European countries. So that's it from my side. If you have any questions, please let me know. And you can also visit our website, acobioscience.eu.
Thank you very much, Despina. Now we will move to contrast project. Steven, hello. Can you take the control and change the slides?
Yeah, good afternoon, everybody. Hopefully I can take control. Okay, yeah. So yeah, I'd like to just talk about the contrast project, which is looking at the effects of chemicals of most concern in the marine environment. This is just focusing on one small aspect of that, which is dealing with one of the case studies which we're using sort of biological effects tools in order to look at the impacts of these chemicals and other chemicals as well within in situ in the real environments. Hopefully you can still see my slides. It's looking quite weird for me.
You can change the slides. No worries. I will move the slides when you want to change them.
Can you see that?
Yes.
Yes. Okay. Apologies for that. Okay. So the objective of this work is to provide an integrated chemical and biological effects monitoring strategy to determine the health status of the marine environment. So basically, we want to target sentinel species in the marine environment, such as fish or mussels or other creatures, in order to look at the health status of those individuals, which then can refer it to the health status of that environment which they're exposed to and they live in. This is sort of a method which is sort of adaptation and improvement of the MSFD guidelines, which is mostly focused on chemical assessment, and also trying to include biological effects assessments within those guidelines in order to evaluate good environmental status. So this is certainly looking at, as a contrast project, is looking at the effects of CECs. So those are our target audience, but we can also realize that the methods that we're applying is relevant for the chemicals in the environment as well. Oops. Okay, so we have 4 case studies within the contrast project, which we're targeting. And today I'll just focus on one of them, just to give you an example of the work that we're doing. So here you can see the Oslo Fjord, Liverpool Bay. There's one in the North Sea, and then one in Salmon Gulf down in Greece as well. So these are the 4 that we've considered that we will look at. But I said we'll just focus on Oslo Fjord on this occasion. Okay, so again, we're looking at collection of sentinel species within the environment. And here in the Oslo Fjord, you can see a map of the Oslo Fjord. You've got the inner fjord here, and there's a sill here dividing the water bodies from the inside and to the outside. So there are very different chemistries and conditions within the different water bodies. Here we're looking at native mussels as one of the target species, where we're collecting them from 5 stations. There are 3 stations. You can hopefully see these yellow dots here within, and this is sort of an urban area. So this is Oslo, and there's lots of there's a sewage treatment plant coming in here and over here as well. And so, and there's lots of boating activity, and there's also runoff from a lot of industrial activity within the area. So this is considered much more polluted here than you would expect further out as you go. But we have additional stations of mussels, the outer fjord near Drøbak, and then one further down in Vala area, down here as a reference location. The idea is to collect mussels, roughly about 100 per group, and look at it and trying to get the same sort of age or size class, so it's easy for comparison. In addition to the mussels, we're also collecting fish. We're trawling for fish within. Again, we're looking at the inner fjord and then the outer fjord areas, and these sort of orange blobs that's there to design. This is the rough area where we trawled within those water bodies, and the target fish species within the fjords were whiting, as pictured here, and haddock as well. And these are often quite demersal fish species, so we consider them to be exposed to sediments, contaminated sediments on the floor as well. Okay. So what we want to apply, and sort of the concept of the project is looking at the mechanism of toxicity of compounds. So we apply this biomarker toolbox, and within the project, this is, like I said, this is in one of the work packages, but an earlier work package is looking at laboratory testing of chemicals of most concern and trying to find mechanisms of toxicity of those chemicals that may be interesting to look at within the field. So this is a field aspect of it, and we're applying this biomarker toolbox, looking at various different mechanisms of toxicity, as you can see here on this graphic, and also measuring several biomarker responses, such as lysosomal membrane stability, for example, for membrane disruption, metabolic impairment. So you've got cellular energy allocation and acetylcholinesterase inhibition, oxidative stress markers, biotransformation, genotoxicity. So quite a broad spectrum of endpoints what we're looking at. What we want to do is measure these in the different target, in different muscle individuals from the different locations, and also link that with the chemicals of most concern that we measure in the soft tissue of these muscles as well. And what part of the project as well is to identify which chemicals of most concern we should look at. And we've identified 64 based on the properties. We have a chemical prioritization scheme which we've published as well to identify which chemicals we've considered to be important and to be measured in our target species. So the plan is to integrate the biomarker responses with the chemical responses to give an overall indication of health status and also whether there's any linkages between chemical exposure and biomarker responses. This is an example. I'm going into a lot of detail here, but this is just an example of how we would sample the mussels within the different stations, where we're using different, like, for example, hemolymph, which is the blood of the mussel, and different target organisms where we will use in order to do some of the endpoints that we require. A lot of the biomarker endpoints require liquid nitrogen for freezing and storage at -80. So there's considerations regarding sampling. But again, we won't go into a lot of detail other than we're looking at biomarker responses within target tissues, as well as looking at CCs. And also, I should say that we're looking at non-target analysis as well that we'll be carrying out on the muscle homogenates. Okay. In addition, so again, we're looking at fish, and again, we're looking at the mechanisms of toxicity in the fish and how these CCs can interact with and cause harm to the fish in terms of endocrine disruption, for example. We have a few endpoints or markers that we're looking at, like, for example, histology, thyroid, VTG responses, vitellogenin, if you're not sure what that is, membrane disruption again, neurotoxicity, cardiotoxicity, oxidative stress, immunotoxicity, biotransformation, genotoxicity, and carcinogenicity. So quite a wide suite, and we have more options in fish as than we did in mussels, and so we have a bigger toolbox that we would like to apply to these case studies. The idea is that we're going to apply them, see the results, and analyze results, and then provide some sort of interpretation or recommendations of which endpoints seem to be the most responsive, so that we could maybe suggest them for MSD guidelines as well. And again, the sampling procedure for the fish is quite a bit more involved. We have blood samples we've taken for different endpoints. So we're looking at whole blood for genotoxic markers, comet and micronuclei. And we're looking at other markers within the blood plasma of the fish as well, and various components what we'll be analyzing as well. Bile, I've mentioned before, we're looking at maybe transformation products. We're looking at metabolites. We can look in the bile, since fish are known to metabolize many compounds. And then liver is a very important target organ for this type of work. And so there's many endpoints that we'll be looking at, including chemicals within the liver too. Additional tissues, looking at gills, gonads, and the fillet of the fish, in addition to the heart with gene expression and supporting information such as age from the otolith as well. So it's quite a comprehensive sampling plan that we propose. So what we're going to do with this data, unfortunately, this is still an early stage, so we don't have the chemical data I can show you, so it's still not available. But what we are planning on doing is to apply it to integrated frameworks. And there are suggested CHASE, for example, which is the CHASE tool looking at chemical assessments, and also this OSPAR IC's traffic light system that we plan on using as well, and maybe others, but these are two we've highlighted so far. And so this is an example of the CHASE tool, and it provides you a general score of whether a certain area would be based on threshold values for the chemicals, and in our case, biological effects, that we'll see whether a certain area is above the threshold levels for the different endpoints. Also, maybe a traffic light system. So here, a lot of the IC's working groups have presented some guidelines to this and done a lot of the work already. But we propose to use this sort of approach where using these threshold values, which is background assessment criteria, which is BAC, and then BAC to EAC, which is environmental assessment criteria. So these are different threshold values for the different chemicals or the different biomarkers. And you can see that anything over red will be classed as harmful. Green is between the two, so there's some sort of compensation mechanism happening, and then blue is considered to be more reference location. So using this scheme and then combining these schemes, you can get an overall score of whether an environment or a location is particularly harmful based on the chemical and biomarker responses. So it's considered more a holistic sort of monitoring tool for environmental assessment. Okay, and I think that's all I had. Thank you for your attention.
Thank you very much, Stephen. Very interesting. If you have any questions from this presentation, don't forget to drop it in the chat. And we will move forward to our last presentation from SUNBIO project. And at the end, we will answer the questions, and that's it for today. Elena, can you hear us?
Yes, I can. I hope you can also hear me.
Yes, very well. You can take the control and move to your presentation.
Yeah, just give me a sec. Thank you. Just a moment.
Yes.
Okay, you can hear my first slide.
Yes. You're
Okay.
correct. Okay. You can
Okay,
move on.
perfect. Okay. Okay, thank you. Good afternoon, everyone. Good afternoon from Greece, actually. First of all, thank you for the opportunity to present in this webinar. And of course, a special thank to Christa for organizing this event. My name is Elena Tilegrafou. I am an archaeologist and I'm working in Atlantis Consulting, an SME company based in Greece, which we are also a partner. We are also a partner of SUNBIO project. So I will briefly present the SUNBIO from a monitoring to remediation perspective, focusing on how environmental data can support not only understanding, but also active ecosystem enhancement. So as you already may know, marine ecosystems today are under increasing pressure due to the climate change, biodiversity loss, and human activities. At the same time, there is a strong need to expand offshore renewable energy as part of the green transition. So this creates an important challenge which SUNBIO aims to address, how to ensure that ocean-based solutions support rather than compromise marine ecosystems. Also, the project goal is also aligned with broader frameworks such as the EU mission Restore Our Ocean and Waters, and also the United Nations Sustainable Development Goals, and of course the Ocean Decade Agenda. A few words about the SUNBIO project. SUNBIO is a European research project funded by the Horizon Europe program. SUNBIO explores how offshore renewable energy systems can be developed in synergy with marine ecosystems. The objective is not only to reduce environmental impact, but to move towards more let's say, nature-inclusive offshore solutions. Just a moment. So, of course, there are some technical developments of SUNBIO. SUNBIO develops an integrated system, and the three main technological components are first the offshore hybrid renewable system. This is a floating platform that produces energy using solar panels, wave energy technologies, and thermal energy systems. Second, we have the underwater biodiversity hub. This is a structure that supports marine life by creating safe spaces where marine organisms can grow. And it works in a similar way to an artificial reef, helping marine ecosystems recover and develop. And third, we have a sensor suite for environmental monitoring. These sensors collect data of the marine environment, such as water conditions and other ecosystem activity. This information helps researchers better understand how renewable energy systems interact with marine ecosystems. In this slide, you can see an illustration of the overall SUNBIO concept. At the surface, you can see the offshore energy platform combining different resources, renewable energy resources. And on the seabed, you can see the biodiversity structures and the creation of the underwater biodiversity hub, which, as I said, function as habitat that supports marine spaces. So as you can see, this is not just an energy system, but an integrated environmental system connecting surface infrastructure with the underwater ecosystem. So from monitoring to remediation perspective, of course, one of the main pillars of SUNBIO is the transition from monitoring to remediation. So first, environmental monitoring provides continuous data on marine conditions and ecosystem activity. Then this data is analyzed to understand how ecosystems respond to the presence of offshore infrastructure. And based on this understanding, targeting interventions could be implemented. So in the case of SUNBIO, this includes the development of the Underwater Biodiversity Hub, which actively support marine life and contribute to the ecosystem restoration. Our system is going to be tested in real conditions in the OBSEA Observatory in June 2026. OBSEA Observatory is the SUNBIO pilot site. This observatory is located about 4 kilometers off the coast of Vilanova in Spain. And this observatory already collects long-term ocean data, which is very useful for the system, as well as it allows us to compare new data from SUNBIO with the existing environmental information. So why this matters, why this approach, the approach matters, what's the positive impact? So the starting point is ensuring that offshore energy systems do not compromise marine ecosystems. Through monitoring, it becomes possible to understand real ecosystem responses, and this creates the opportunity to move beyond impact assessment towards action, towards active ecosystem support. And finally, this contributes to a more sustainable and resilient ocean use, let's say. So this is not a linear process, is let's say something like a continuous cycle. Let me show you a short video, a short presentation of the SUNBIO, and this video is also uploaded on our YouTube channel. So I'm starting playing the video. Before ending this presentation, I would like also to invite you to the upcoming SUNBIO workshop on Posidonia Oceanica restoration, which will take place on 3rd of June. This is going to be a hybrid event with both online and on-site participation. It will take place in the Universitat Politecnica de Catalunya campus in Vilanova. So you can find more information about the workshop and the registration in our website, sunbioproject.eu, or scanning this QR code. I think that was everything from me. Thank you very much for your attention and for being here until the end.
Thank you very much, Elena. Very interesting. And also don't forget to register for the workshop. I'm sure that it will be very interesting.
Thank you for
And now I will stop sharing the screen and share my screen for the Q&A session. Let me share my screen. You can see the questions?
Yes.
Yes, we
Perfect. For the first one for iMermaid project, Jasmine, are you here with us?
Yes, I'm here.
Perfect. What was the main challenge you faced when implementing your solutions?
Yeah, maybe it was not very clear from the presentation because I tried to be in 10 minutes, so I just mentioned them. But we faced mainly 3 different challenges for the heavy metal box, which is a high electrical noise in the signal that was not present during the tests that were performed in the laboratory of the University of Florence, because in an industrial site there are a lot of, I don't know, for example, pumps, some control units that are connected to the electricity. So this caused a very noisy signal, and to reduce this signal, we had to change the acid protocol, acidification protocol for the pretreatment of the sample, moving from acetate buffer solution to hydrochloric acid in order to enhance the conductivity of the solution inside the cell. Then we also grounded the instrumentation and purchased a noise filter that was attached to the instrumentation to reduce the noise. Then we also faced another challenge that was the formation of air bubble inside the measuring cell, and this we think was due to the fact that there was some oxygen dissolved in the water, and these micro bubbles, they can just they merge together to create bigger bubbles that were formed inside the cell, and this prevented the connection between the reference cell and the working cell. So in order to reduce the content of dissolved oxygen in the water, we purchased an additional buffer tank in order to let the water stay there for 2 hours prior the analysis, in order to give time to the dissolved oxygen to be released in the atmosphere and reduce the air bubble formation. And with this approach, using hydrochloric acid, we were able to monitor with sufficient sensitivity 3 out of 4 heavy metals. The last one, which was zinc, we have to change again the pretreatment protocol of the sample, the acidification, going back to the initial protocol using a buffer acetate. So we had 2 different protocols, one with the hydrochloric acid for cadmium, lead, and copper, and one with buffer acetate for zinc. While for the Furdy scavenger technology, which was the removal technology, once the capacity of the filter is filled, you have to wash and regenerate this filter. What happened during the pilot is that we have experienced more high pressure than foreseen. This was due mainly to a not very proper prefiltration system that were implemented for the pretreatment of the water. And secondly, when the system was shipped back to Finland, they the technology provider opened the unit and noticed that the filters were they were broken during the transport by truck from Finland to Italy. So this was the 2 main reasons of the high pressure that we experienced. And this is the reason also why they did an additional test in Finland, both in laboratory and in another industrial site in Finland. I hope I replied to your question.
Thank you very much, Jasmine. And we have two questions for remediation project. Which kind of photobioreactor design you use? And the next one, have you observed efficiently elimination of PFAS?
Yeah, so for the first one, the photobioreactors, this is actually a patent from a company in Portugal called Blue Matter. That is actually they designed these photobioreactors. So as you maybe saw the image, these photobioreactors is kind of like a cube, but then one of the sides is inclined because this is to maximize the growth of the algae. So they have more surface for sunlit, and then also within that they put kind of like a bubble curtain, so then it prevents the attachment of algae into that side. So then it will potentially create biofilms and so on to reduce the penetration of sunlight. So this is from the Blue Matter. So I'm happy to also, if you send me an email, it's on my email contact is on the slides. So I'm happy to put you in contact with the main contact for this in the project. So have you observed efficiently elimination of PFAS? So it's just a short question, but I have like different components. So have you observed elimination of PFAS? Yes. It's on the policy brief that, for instance, one of the test sites, the maximum removal efficiencies were from 50% to more for PFAS that were measured. But then, of course, for the efficiently elimination of PFAS, as you must consider, this project was testing the technology, so there was also a lot of challenges for the implementation. So the water had to be cut off. There has been also clogging and so on. So the thing is that while their couple of months were tested, there is not a constant say like, okay, it's fully operational, because there were a lot of operational problems. So we cannot say that has been efficiently eliminated, but it has the strong potential to be efficiently elimination of PFAS. Of course, it depends which PFAS are you measuring and so on. Yeah, also again, I will be more than happy to put you in contact, because I know there is a deliverable highlighting this, and of course I'm just presenting the policy brief.
Maxime, you raise your hand because these questions
It's
were
my
yours.
question, yes.
So.
I work in IMAMED project, and we measured PFAS, particularly PFOA and PFOS. But have you focused on others one or usual one, PFOA, PFOS, and at which level concentration have you measured them? So this is a complementary question. Sorry.
No, that's good. And unfortunately, I'm not able to reply to that. Oh, sorry, I realized I don't have the camera on. Sorry, I cannot able to respond to that. But if you send me an email or to the Luca, maybe you are the coordinator, he will be more than happy, or myself, to put you in contact, because indeed it's quite useful to exchange this kind of.
I have sent to you on LinkedIn a demand of friend to be a friend with me. Okay, we can change by that way.
Yeah.
Thank you. Bye-bye.
Thank you very much for this collaboration. And two questions for Angelo. What are the main advantages and limitations of using natural bioreceptors comparing to bio-inspired ones? And the next one, does the histogenic sensor quantify estrogenic mimicking compounds in addition to this one? And this question was from Steven from Contrast Project, so you can answer them.
Okay, thank you both for the questions. So to reply the first one, what are the advantages of bioreceptors? The advantage is that they are super specific. We can assume that they have been selected for the target molecule for millions of years, because basically we retrieve them from living organisms, so they have been selected to work very well. But unfortunately, they have also the limitation that you need, for example, antibodies or receptors or ligands that we can find in any kind of cell, both human bacteria or other kind of animal. You have to somehow cultivate them, you have to extract, you have to purify. So these are very energy intensive methods, and the cost is very high. So they are good, the bioreceptors are good because they have high specificity, but to produce them, it's very expensive, and then you have to use cells-based mechanism to produce them. Bio-inspired, usually they are synthetic. So you use what is called solid-state chemistry. You take one base or one amino acid each time, and then you attach, and to a certain length. For example, you can easily synthesize peptides around 15 nucleotides, or DNA molecules around 70, 80 bases. So, and this is the size that usually it's enough to have bio-inspired receptors. They work well, and what you don't need any living organisms, and any companies in the world have the same standards of production, so it's much better. For the second one, thank you again, Stephen. So yes, I'm afraid that we will have this kind of, let's say, cross reaction with other estrogenic or non-estrogenic. Even testosterone, we may have it. We didn't test it, but the beauty of using the first electrode that I showed you, I don't know if you remember, there was a sort of curve with a peak. So when estrogen interacts, it interacts at a specific peak. If, for example, another kind of estrogen molecule that could be testosterone, for example, or the oxidative form of estrogen interacts with aptamer, this peak shifts a little bit. So electronically, we can filter this kind of noise, and we can somehow separate the cross reactivity with other species. For the second one that I showed, the one that has just some dropping curve, we cannot do that because we just see a standard signal that drops. But for the first one, we can somehow do it. But yes, we have this problem and we have to solve it.
Great. Thank you, Angelo.
Thank you.
Okay. That's all for today. Thank you all very much for participating, for presenting, for everything. I hope to see you all soon. But the Mermaid project, it comes to an end at the end of May. But we will continue to support each other in any way. We will be in contact, and I will share with the presenters the recording. So if you want to publish them or upload on them YouTube, I don't know. So thank you again all very much, and we will be in contact.
Great. Thank you. Bye-bye.
Thank you.
so
Thank
much.
you.
Bye-bye.